You probably think it's easy to tell the difference between a plant and animal when you're looking at organisms on this scale, but can you tell the difference between plant and animal cells on the microscopic scale? This is comparing plant and animal cells. The skill of distinguishing between types of cells on the microscopic level is useful in many different professions, including forensic science, food quality control, and pathology, or the study of the cause of disease. As you look at plant and animal cells in the first two parts of this lab, apply what you've already learned about plant and animal cells to what you see on the screen in SPARKvue. For example, you learn that plant cells have rigid cell walls, while animal cells have flexible cell membranes. Let's get started. I'm using a Windows computer with SPARKvue. You could use a different device as long as it has a USB input and a SPARKvue compatible microscope if you're doing this lab on your own. I'm using a compound microscope, but a single lens microscope could also work. There are some models that connect via Wi-Fi, but we're connecting through USB today. The USB cable should be unplugged from the device before you start, and because some apps will grab the microscope as soon as it's plugged in, make sure there are absolutely no apps open on your device. And now we're ready to begin. There's no actual data collection in part 1. This part of the procedure is all about setting up the microscope in SPARKvue. Make sure the SPARKvue app is set to full screen and choose sensor data. SPARKvue is seeing the built-in camera on this laptop as camera 1, so while I'm looking at this screen, I can plug in the microscope's USB cable. And now I can see a new camera join the list. Camera 2 is probably the microscope, but if it isn't, I'll show you what to do in a moment. So now I need to open the camera 2 menu and check the image checkbox to get access to the video feed. Once that image box is checked, a camera option appears in the templates menu. Click the camera icon to open a live camera feed in SPARKvue. If the live feed is not coming from the microscope, click inside the image to stop the video stream. Open the camera tools menu. Open settings. Try a different camera. Then select preview mode to go to live stream mode. We're going to view wet mount slides with stain, so the best lighting choice is the source below the stage. And now we're ready for part 2, where we look at animal cells. In this part, we're going to look at animal cells donated by me, a mammal. First, I'll grab a clean glass slide and cover slip by the edges and inspect. I'm looking for a chipped or cracked glass, but these look fine. If it's chipped or cracked, I need to discard in the broken glass container, but I'm just going to set these on the paper towel since they're not chipped or cracked. Now I'm picking up a clean, flat toothpick by the narrow end so I can use the broad, flat end to gently scrape the inside of my cheek with an up and down motion to collect a sample of cheek cells. I'll do a set of 15 up and down scrapes inside both cheeks. Now I'll smear the flat end of the toothpick in the center of the slide over an area no wider than my thumb for 3 or 4 seconds. Flip the toothpick over and repeat. Throw the used toothpick in the trash. I'm preparing a wet mount slide with methylene blue stain. Whenever you prepare slides with stain, use a lab coat and gloves if available. I'll add 2 drops of methylene blue stain over the smear. Not too little and not too much liquid because there should be just enough stain to cover the area between the cover slip and the slide with little or no extra liquid escaping from under the cover slip once I place it. I'll set the cover slip at an edge, on its edge, near the stain like this. Then when I'm anywhere from 1/2 inch to 1/4 inch above the drop, I'll release the cover slip So it gently falls on the drop, and that helps avoid trapping air bubbles. You're supposed to let the stain spread over the area between the slide and cover slip on its own, and you should not push down on the cover slip. Use a piece of paper towel to draw excess liquid away from the cover slip without touching or pushing on the cover slip. Just a little bit there. And I got a little bit on the outside there. Okay, we go to the other side. I'll slowly inspect multiple areas of my selfie at multiple levels of magnification, and I'll just lighting and focus as I go. But I can't do that yet because part of the observations you need to record on your answer sheet includes magnification power. And since we're using a compound digital microscope, there are two magnifications to consider. First, there's the magnification power just looking through the microscope optics. The eyepiece has 10x magnification, and there is a 4x objective, 10x objective, and 40x objective lens. I won't be using the oil immersion lens, so we'll skip that objective lens. Remember to multiply the eyepiece times the objective to get full microscope magnification power. So when I'm on the 4x objective, the total microscope magnification is 4x times 10x, which equals 40x total magnification. Then there's the digital image to consider. You have to figure out the digital magnification on the screen for each microscope objective size. You need to calculate digital screen magnification by taking the actual millimeters across the screen as measured by a ruler and dividing by the millimeters as measured through the microscope. I'll use a micrometer calibration slide to get the microscope distances. Now let's look at the slide with the 4x objective lens. Once I have the micrometer calibration slide in focus, I can see these measurement lines that are printed on the calibration slide. From one end to the other is 1 millimeter. So I can calibrate SPARKvue's measurement tool. Now I have to figure out the precision of this slide. So if the distance is 1 millimeter, these longer lines are 10th millimeters. And you can't really see the small lines really well, so I'm going to zoom in and show you there's even smaller lines on the slide. There we go. So these smallest lines are 100th millimeters. I get to estimate one more past the last marked measurement, so that means I can read to the 1000th millimeter. That's 3 decimal places to the right. So that's the precision. Let me go back to low power. Get back in focus. So we can do the 4x objective lens. Okay. Now I have to click anywhere on the live image to capture a still image. Then I need to open the measurements tool and click this ruler up here and click anywhere inside the image capture. I'm going to click in the center of the X to draw out an arrow and drop it on 0 millimeters and move the other end to point at 1.000 millimeters. I'm going to click inside one of the arrow boxes to open the image measurement properties menu. So the known length is 1 millimeter. I can set digits to 3 since the slide is readable to 3 decimal places, and the units are millimeters. And click OK. And now the digital ruler is calibrated. So I need to get SPARKvue's measurement of that video area from edge to edge. Let's see. I want to go from exactly the edge of the video feed or video still where it meets the white area of SPARKvue, edge to edge in a straight line. Okay, SPARKvue's measurement of the video area is 2.279 millimeters across. I'm going to write that down. SPARKvue is 2.279 millimeters across that area. And now I'm going to move SPARKvue over to this other screen so you can see it more easily. So SPARKvue measures 2.279 millimeters across, but clearly this arrow itself is bigger than just a couple millimeters. So I'm going to grab the ruler, and I need to use metric and remind myself that this is actually centimeters. Oh, I should probably look at the precision of this ruler. If I'm looking, I see lines down to, I'm working in millimeters, right? So I see only 1 millimeter lines. That means I can only estimate to the 10th millimeter, so it's not as good precision here. So I can see in centimeters, this is 20.39 centimeters. Multiply that by 10 to get it to millimeters. So it's 203.9 millimeters. Gotta write it down because it's hard to remember all these numbers. The magnification is the metric ruler distance divided by the digital ruler distance. So on my screen, every time I use the 4x objective lens, the magnification is, let's see, what is it? It's 203.9 millimeters divided by 2.279 millimeters. And I'm going to round that to a whole number. And I get 89 times, about 90. So 89 times every time. I'm looking at 89 time magnification on this screen. If we think about the optics, it's 40 times magnification through the scope, 89 times on the screen. This is why we have to do magnification power for each thing we're looking through. So now I'm going to move SPARKvue back to my computer screen. I'm going to set up slide snapshots for the 10x and 40x objective lenses as well, so you can calculate the magnification on your own. You will need a ruler, so take a moment to pause and figure out the magnification on your own screen as we go. Make sure you've got SPARKvue open to full screen before you start, and make note of the screen magnification in the margin on your answer sheet as you figure it out. So I'll turn on the preview in SPARKvue, then switch to the 10x objective. Oh, first I have to make sure I've got my previous capture in focus. There it is. I'm going to center the image, and then I'm going to switch to the 10x objective lens and focus. Switch to fine focus now and recenter my image. So here on the 10x objective, I have to calibrate to the 10th millimeters. Remember those long lines, the 10th millimeter lines. Since I can't see the whole millimeter anymore, I'm going to draw a measurement line across the 10th millimeter. Take my capture. And I'll set the known distance to 0.1 millimeters because it's a 10th millimeter. But still keep the precision of 3 digits. Go to millimeters. Okay. Now on your side, you have to set the arrow to go across that microscope view from edge to edge like I did before in a straight horizontal line. Then you have to grab your ruler and measure the arrow point to point, convert to millimeters. Finally, take that ruler distance in millimeters and divide by the SPARKvue arrow distance. It should be bigger than the last one. You're going to round your answer to a whole number and make a note of the screen magnification for the 10x objective lens on your answer sheet. Finally, the 40x objective. Let me turn on preview in SPARKvue. Switch to the 40x objective. Focus the slide. And go to my fine focus. And I can still, looks like I can still do 10th millimeter like before because I still have those long lines in view. As long as I can see two adjacent lines, I can do that. Take my capture. The length is still 10th millimeter. And now you have to figure out screen magnification on your end and make a note of it for the 40x objective. Now back to the animal cells. Pause the video each time I get a section of the slide in focus so you can take time to examine many different cells and identify as many cell structures as possible. Look for the cell membrane, cytoplasm, and nucleus. When you see a microscope view you think you'd like to add to your answer sheet, look for the snapshot number in the legend so you can locate the same snapshot in the SPARKvue sample data file. So right here, the one I'm looking at now is number 3. Remember to click on the legend to switch between snapshots and go back to 3, 2, or 1. Okay, so starting with 4x objective, go back, and let's look at my selfie. Turn on preview in SPARKvue to get a live image and search for cells. There they are, they're all over the place. And those yellow circles you see, that's actually trapped air bubbles. I got a few of those, and it took us so long to do those calibrations that it dried out a little bit, but that's fine, it's still good. So you can ignore the yellow circles. They are trapped air. There's more than just cells here. There's also some food particles, and I'm embarrassed to say, a little bit of bacteria. I did brush my teeth. And I'm going to find a nice few cells to look at. I'll just take the snapshot. We're going to have very few air bubbles, but lots of cells. So let's look at this one. I like this one. So there is some lint here. You can ignore that lint. This is an air bubble. You can ignore that. So we're looking at the blue things. These are the cells. And when you see clumps of blue, that's just a bunch of cells all clumped together. So you really want to look at individual one on their own cells. So I'm going to take my snapshot. And now I'm going to start preview again so that we can look at the same cells at a higher magnification. Now I'm on the 10x objective lens. I'm going to focus. Coarse first and then fine. There they are at higher magnification. Check those out. You can probably see those cell structures a lot more easily. I like this view, actually. I'm going to take my snapshot just right here. This is the 10x objective lens for image number 5. Now I'm going to switch to the next objective lens. Start the live view and 40x on the objective. And let me magnify. Oops, that was close. Go to fine focus. So let me get a cell in view here. You'll notice that because cells are three-dimensional, if I slightly change the fine focus just a little bit, you'll notice some things go into focus while others get blurry. That's because cells are three-dimensional. See that? So let me do a little bit of that for you. And remember, you can always go back and pause if you see something that you want to capture. And I'll move around so you can have a few views of cells at a higher magnification. There's some more. Okay, I'll move more. Look at more this way. Okay, and just to remind you, this was methylene blue stain. That's why they're blue. They would be clear if you did not use the stain, and a lot harder to see the cell structures or cells themselves. All right. And I am going to take a snapshot in a moment. Let's just go back to where we were a second ago. Or no, in fact, let's find new ones. Okay, and that's the 40x objective. You'll need to add annotations or notes to the snapshot you choose so you can point out visible structures. First, you turn on that annotation tool, click on the structure you want to label, type the structure name, I'm just making up a random name, then click OK. Click and drag the box to move the text annotation. You can also move the arrow if you click and drag the pointy part of the arrow to point to something else or somewhere else. Add as many annotations to an image as desired. To remove unwanted annotations, open the annotation tool, select the eraser, and then select a label to remove. Before moving on to part 3, choose one captured image at 1 magnification and complete the row for animal cells in table 1 on your answer sheet. Check with your instructor to see whether they prefer captured images or sketches or both, and go online to look up how to do an image capture on your device if you're not sure how. Include labeled cell structures whether you use a screenshot or a sketch in table 1, and don't forget to record notes and observations, including microscope and screen magnification powers. See you in part 3. Now we're going to look at plant cells, courtesy of this onion. The scalpel and forceps will help me gently separate the outer skin, which is very thin, nearly transparent, and is very flexible. Oh, I had an extra layer of onion on there, so peel that away. And get the blade underneath the purple part of the skin. That's enough. Now, once the skin is separated, it should easily peel off. I'm trying to get a single layer here. Drying out already. Now I've got an onion skin sample that is about or less than 1/4 of the size of a cover slip. It's actually like 1/8, but that's fine. Please remember, whenever you cut anything, just be sure to work on a solid surface and always cut in a direction away from your body. So set the sample in the center of that slide. Okay, I'm kind of worried about the tail here, but it's thin enough that I can just get the cover slip over it. I just want to make sure I don't have too thick of an onion skin on there. I should have a single layer if I can. We're going to prepare this wet mount with iodine. I'm going to use a few drops. Looks like it took 4 drops. Set the cover slip like before. Okay. And I'm going to use that paper towel to sop up just a little bit of the extra iodine on the edge there. So again, I'm going to take snapshots at 4x objective, 10x objective, and 40x objective lenses. So you can choose one in the sample data file to annotate with cell structures, and you will complete the row for plant cells in table 1. Look for the cell wall, cell membrane, vacuole, nuclei, and cytoplasm. So I'm going to start at 4x. Okay, wow, we're looking at a whole lot of colors here. Let me focus. Aha. This is very interesting. So on this area over here, this is just light coming through an air bubble. So we're going to ignore the blue part. This is some onion skin that's folded over on itself. So it's not good to look here because I got more than one layer. I'm trying to find a single layer here. This looks like a good area to explore. Let me move over in there. So what I'm going to do is focus and take a snapshot for you. There's some air bubbles on the left side there. Let's see, which side do I like better? I like the right side better, so I'm going to focus on the right side there. Here's the image at 4x, and then I'm going to collect 10x and 40x objectives for you. I'll add that to the sample data file, and I'll see you in part 4. Now for the challenge. I'm going to share 3 more slides, slide A, slide B, and slide C. You have to look for clues to figure out whether each shows plant cells or animal cells. There's a row for labeled images and observations for each slide in Table 1. All the slides will show up in your sample data file. I've renamed each image to match the letter and objective lens I used. Right now, image number 10 is slide A with the 4x objective lens. When I click the legend, I can scroll down and choose the 10x or 40x versions of slide A. Then slide B at the 3 objective lens powers, and slide C with the 3 lens powers. Fill out Table 1 as you go, then answer the questions at the end of the lab. Good luck!